introduce yourself, where are you from, and what do you do?
I'm Mario Ventura. I'm from the University of Bari, Department of Bioscience, Biotechnology and Environment, and I'm a geneticist. And I work on and look at chromosome and chromosome evolution in human and in primates.
Perfect. Here we can see researchers at the Sanger Center. It's 1997 and they are racing to finish what would be known as the Human Genome Project. Now there's no doubt about it, the Human Genome Project was one of the most ambitious scientific efforts in history. For the first time, scientists attempted to read the complete genetic code of a human being. It was an extraordinary achievement, but these were still the early days. Here's one of the leaders of the center, Sir John Sulston.
The discovery of the sequence, the code of life, is only the beginning. It's very important to realize that it's not the end of the process, it's only the end of the beginning.
First time we sequenced the chimpanzee genome, what we did, just to simplify the work, was to remove all the repetitive elements in the genome, including one of the most important region that we didn't know at that time, that were the segmental duplication. Now we have the technology, we can take in account the more complicated region like the centromere, like the segmental duplication, and neocentromere. So centromere is defined as the primary constriction on a chromosome. So if you just have in your mind how a chromosome is, you just keep in your brain the image of an X. The centromere is the center.
So imagine you're on holiday and you're bringing with you 2 suitcases. You arrive to the airport, you do your check-in, they weigh your bags, and the lady at the counter gives your bag a label. Now imagine it's a barcode with 178 numbers repeating over and over and over again. Now your suitcase needs this label because when it comes to the automated baggage system, machines are going to read that label, they're going to grab the bag by the handle and move it to the correct conveyor belt. Without that correct label and suitcase handle, there's a big chance your suitcase is ending up in Timbuktu. Inside our cells, centromeres operate in a surprisingly similar way. Before a cell divides, it first has to make a complete copy of all its DNA. Then, tiny structures called spindle fibers attach to the centromere and physically pull the chromosome copies apart to opposite sides of the cell. It's an incredibly precise process. And when this process fails, cells can gain or lose chromosomes, contributing to cancer and many other serious diseases. But things get even more complicated during the production of sperm and egg cells. Unlike normal cells, these cells must divide twice during a special process called meiosis. First, the chromosomes inherited from your mother and father pair together and exchange small pieces of DNA in a process called recombination. After all, you are not an exact copy of either parent. You are a unique mixture of both. The cells then divide once, and then they divide again. And by the end of the process, sperm and egg cells each contain 23 chromosomes ready to combine during fertilization. Throughout each stage of this process, the centromere is essential. Because if chromosomes fail to separate correctly, even once, genetic information can be lost, duplicated, or inherited incorrectly.
If a centromere is not working properly, there are mistake errors during segregation, and so gametes can be created as unbalanced gamete. For example, the most common trisomy in human is known as trisomy 21, that is the cause of Down syndrome. Actually, this is caused by a mistake in segregation of one on chromosome 21. So knowing the centromere function, knowing what is wrong when the centromere doesn't work properly is critically important for human health and human genetics.
So here's the bottom line. The centromere is incredibly important for cell division. One small error in chromosome segregation can have enormous consequences. So you would expect something that's this essential to be evolutionarily stable, fixed, conserved, untouchable. But this is where things start getting strange. Now take a look at these chromosomes. From horses to donkeys to zebras, horses have 64 chromosomes, donkeys have 62. That alone isn't especially surprising. They are a different species after all, but the chromosome number isn't the only thing changing. Look closely at the centromeres. Their locations are different as well, which means evolution isn't just reshaping chromosomes, it's reshaping the necessary structures responsible for chromosome segregation itself.
There is a huge differences between species, and I can add you also inside the individuals. Actually, you know that we have 46 chromosomes, 23 come from our mom and the other 23 comes from our dad. We did the comparison between the two homologous chromosomes. The centromere coming from the mom and dad are completely different.
And that raises a huge question. How can something so essential, so precise, so delicate and necessary also be so dynamic? How can the centromere become a force for evolutionary change? To understand that, we first need to understand, how does the centromere move at all? So I want you to think back to the suitcase metaphor. Imagine your luggage tears open halfway through the airport baggage system. Clothes spill out everywhere. Ideally, you'd want the suitcase perfectly repaired, but in emergency, the airport might do something much simpler. They would tape it back together, strap on a new handle, and send it on its way. Is that solution perfect? Not necessarily. Some things might still get lost, some things might fall out, but it's a far better version than losing the entire suitcase altogether. And neocentromeres may work in a surprisingly similar way.
So, neocentromere represents area of the genome or chromosome that activated and are really important for the segregation of the chromosome that have not the classical organization, this 171 base pair that I mentioned before. So they are totally avoided of this sequence, but they still keep working as a great functional centromeric region. Mostly the first analysis that we had the possibility to do was on clinical cases, because they could be harmful in an individual, because when a neocentromere is formed, it's usually formed after a chromosomal breakage. So this small piece, instead of being lost, is recovered by the activation of neocentromere, and this creates an imbalance, gives to the individual a pathology, a phenotype, because there are more genes that instead should not be there.
Neocentromere can form when the original centromere becomes inactive. Now, so far we've talked about the most obvious situation. Maybe there's a chromosome break or the original centromere gets damaged. But that's not the only way this can happen. Sometimes a neocentromere might appear simply because of a mutation, or maybe because of epigenetic changes. And honestly, scientists still don't fully understand why certain regions suddenly become neocentromeres. In fact, that's one of the big questions Mario Ventura's lab is trying to answer. But he's also tried to understand cases in which neocentromeres appear fully compatible with healthy development.
On the other side, there are other cases that we found where neocentromere are not harmful. So centromere can just change position, and we found a person that had this centromere that has changed position, just moved from a regular position to another position, and it worked perfectly as a centromere.
Sometimes these neocentromeres don't seem to cause any obvious problems at all. Sometimes they remain completely stable, and they get passed on through generations, and over long evolutionary timescales, they can eventually become permanent new centromere locations, what scientists call evolutionary new centromeres.
We are working on this direction because now we have the possibility, we found orangutan cell line that are two species, and these two species have in their wild population 20% of them have this neocentromere. So exactly the case that we have for human, we have in two species, two orangutan species. We are still investigating on this aspect, because if you can find something that can trigger the activation of a neocentromere, we can explain anything related to the centromeric function.
It's incredible to see how far we've come. What was once largely ignored during the Human Genome Project has now become one of the most fascinating areas of modern genetics. Centromeres are essential for keeping our chromosomes stable, and allowing cells to divide correctly. But at the same time, they also seem to be surprisingly flexible, capable of shifting and changing across evolution. These tiny shifts in centromere position, passed on across generations, may eventually contribute to differences between populations, species, and in many ways, life depends on this delicate balance between stability and change, and the centromere may be one of the clearest reflections of that relationship. Hidden deep within the centromere, Mario Ventura's lab may be watching evolution happen in real time.